Hello, everyone. In today's episode, we are going to talk about human versus animal models of behavior. So, let's talk about the different kind of strengths and blind points of animal models. We have often almost known in behavioral neuroscience that animal models are the prefront of any experiment, so to say. This is because it's much more practical, it's easier to observe in a simpler system, but we are not quite uh, we do not have much information about human models of behavior, which has been pretty recent in comparison to animal models. So today with us is Garima, who is going to give us all her insights on this topic. So without further ado, Garima, you tell us about let's open the stage with talking about strengths and the blind spots of animal models.
Okay, so hello everyone. I would try my best to give you the insights on this topic as far as possible. So, I think before discussing this, it's very important to point out that one of the most common questions in behavioral neuroscience, and honestly, one of the most uncomfortable ones, is this that how well do animal models really capture human behavior? I think anyone working in psychiatry or behavioral neuroscience or any field of science eventually runs into this tension. On the one hand, animal models have taught us an enormous amount. But on the other hand, many treatments that work beautifully in animals don't always translate to humans. So, in this episode, I want to think through the strength and blind spot of animal models, not to criticize them, but to understand where they genuinely help and where we need to be cautious. To start with, I think it's important to say this clearly and loudly that animal models are not meant to replicate human psychiatric disorders. They just can't replicate because a mouse doesn't experience depression the way a human does. It doesn't have language, societal identity, or personal narrative. However, and this is crucial, animal models are extremely good at something else. They allow us to study specific behavioral and biological processes in a controlled way. For example, threat learning, avoidance, reward sensitivity, stress responses, and habit formation. These are processes that are highly conserved across species. So, in a way, the real strength of animal model is mechanistic clarity. We can manipulate circuits, genes, or environments and directly observe behavioral consequences. That's something we simply can't do ethically or practically in humans. And at the same time, this is also where the blind spots appear. Because when we stretch animal behavior to stand in for complex human symptoms like guilt, rumination, or suicidal ideation, the model starts to break down. So the key issue isn't whether animal models are good or bad, it's whether we are asking them the right questions or not.
Right. So, I would want us to discuss about, bridging the species gap, like endophenotypes and behavioral domains. So, what do you have to say about that in specific?
Okay, so this tension is exactly why concepts like endophenotypes, intermediate phenotypes, and behavioral domains become so important. Instead of trying to model entire disorder, researchers started focusing on components of behavior. For instance, rather than modeling depression, we might model reduced reward sensitivity or increased avoidance. And rather than modeling PTSD, we might look at exaggerated threat learning or impaired fear extension. These behavioral domains are, measurable in animals, measurable in humans, and often linked to specific neural circuits. So, in a way, we can say that they gives us a shared language across species. Now, what I find interesting is that this approach also shifts how we think about diagnosis. It nudges us away from rigid categories and towards dimensions of behavior, which honestly feels much closer to how psychiatric symptoms actually present in real life. So instead of focusing animals to mimic human disorders, we let them inform mechanisms that cut across diagnosis.
Right. So, when animal models predicted human outcomes and when it didn't, let's have a very broad discussion on that.
Okay, so there are definitely many cases where preclinical behavioral work translated well to humans. Usually, you see where animal models predicted human outcomes. These success shared a few features, such as mainly three features we can say. The first one being the behavioral task was tightly defined. Second being the underlying circuit was conserved across the species. Third and the last one being the outcome measures was comparable across species. So these features were present where animal models predicted human outcomes, and in contrast, many failures happened when animal behavior was overinterpreted or a task lacked clear human analogs, and also when the model focused on face validity rather than mechanism. For example, a task that looks like despair in animals might actually reflect changes in energy, motivation, or learning, not mood in the human sense. So when translation fails, it's often not because animal model are useless. It's because the conceptual mapping wasn't precise enough. If we really want the animal models to be successful, we really need to work on that conceptual mapping and the parameters that are being measured. In a way, we can say that a successful translation depends less on the species and more on how carefully behavior is defined and interpreted.
Right. So, in continuation to this, I wanted to ask you, have you decided that, if given a chance, that which model system you would want to specifically work with?
Okay, so this brings me to something I think PhD applicants worry about a lot. Which model system should I choose and how should I decide it? People often come up with questions like that after designing the experiment. So there are many model systems such as mouse, rat, zebrafish, non-human primates, or human cohorts, and each has its own strength and own weaknesses. And what I have realized is that there's no universally better model. What really matters is the fit. For example, um, rodents are excellent for circuit level and genetic questions. Similarly, zebrafish are powerful for development and high throughput screening. And non human primates are valuable for complex cognition and social behavior. And for subjective experience and clinical relevance, human cohorts are the most essential ones. So, when arguing for a model system, the strongest approach isn't to claim superiority. It's to clearly say this model is appropriate for this question, and here's what it cannot capture. In my opinion, that kind of honesty actually strengthens a proposal rather than weakening it.
Wow. Again, a very, very well rounded answer, I believe. So, you can give your last closing thoughts and then we close.
Okay, so at last, I would like to conclude this podcast by saying that for me, thinking about human versus animal model has been less about choosing sides and more about understanding boundaries. Animal models are incredibly powerful when we respect what they are designed to do. They become misleading when we expect them to explain everything. As someone interested in behavioral and neuropsychiatric neuroscience, I am drawn to this middle space where animal work informs mechanism and human research grounds interpretation. And I think that's where the most meaningful translational progress happens. So, thank you very much for listening.
Thank you so much for being our guest, Garima. That was again very, very insightful, very, very informative. So, thank you so much. That brings us to the end. Thank you, listeners.